Hey guys, this is Kendall Terry, and this is looking at cells, specifically cell organelles in this lecture. So the last lecture we left off by looking at cells and the fact that those cells are made up of 5 particular things, and we talked about cell theory, and we talked about some similarities between cells, but now we're going to look at the actual organelles that make up the cell. So we first look at mitochondria. The mitochondria, every eukaryotic cell has a mitochondria. Even the plants, although that's a common misunderstanding, many people get involved in photosynthesis and they forget that plants also have mitochondria to do cellular respiration, because mitochondria is the cell's energy center. It turns food chemical energy into ATP. If your cell does not have mitochondria, it cannot make ATP energy from the sugar, and that's why it's important to understand that even with plants, that they have a mitochondria to break that sugar that their chloroplasts help make down into ATP energy. Otherwise, they would make sugar with their chloroplasts, but have no way of turning that sugar into ATP. So the mitochondria is what does that. The mitochondria is sometimes called the powerhouse of the cell. That's probably what you've been taught, but we need to go to a little bit better of a definition than that now and know that that is the cell's energy center, where it turns chemical energy into ATPs. Then we have the Golgi Apparatus. Now the Golgi Apparatus, it has this messy look to it, and the reason is because it's this series of membranes, these series of sacs that stuff will come in at the bottom side here, and it does have a particular side that it receives packages from, so to speak, is the bottom side. Then as it goes from layer to layer to layer through the Golgi Apparatus, it will be modified into whatever it needs to be modified into, and then will exit the Golgi Apparatus on this top part, and it does have a specific top side to exit. So the Golgi Apparatus processes, packages, and secretes proteins that the cell has made. It's comparable to a factory or post office that is, if you're mailing a package, you would take it to the post office. They're going to help you put the stamp on it, help make sure they know where it's going, process information, and then send it there. That's what the Golgi Apparatus does. Another organelle is the lysosome. Lysosomes contain digestive enzymes that break things down. Sometimes they're referred to as suicide sacs because they do contain enzymes that help in cell death. And whenever a cell has been given that program sequence, the lysosomes are actually the ones that will go throughout the cell and release these enzymes to help kill off the cell. But they also contain other enzymes. Sometimes it's enzymes to help in digestion outside of the cell. Sometimes it's stuff involved in digesting just something in the cell that needs to be done away with. There's various functions. The next is the endoplasmic reticulum. Now, you don't want to get the endoplasmic reticulum confused with the Golgi Apparatus because they look similar, but there are some key differences. The first is looking at the ones that are called rough endoplasmic reticulum, and that's this first area here. And you can see if you look closely that they have these little dots all over the surface. Those are ribosomes. So this one is involved, the rough ER is involved in making proteins and helping transport those proteins throughout the cell, especially the proteins that are either going to be in the membrane of the cell or get sent out of the cell. They're made right there in the rough ER. The smooth ER is down here, and it does not have those ribosomes connected to it, and it's involved in more of, sometimes it's compartmentalizing and storing stuff. The cell uses the rough ER many times to kind of like as a closet to put certain things in and then release it at different times. Other times it's used to break down, detoxify substances, store certain ions that the cell needs for later, various things. But the endoplasmic reticulum as a whole is involved in transport, intercellular highway that helps move things throughout the cell. Ribosomes are positioned along the rough ER. Proteins are made by the ribosomes, enter the ER for transport to various places. Then we've got the cytoskeleton. Now the cytoskeleton functions in support and contains many microfilaments and microtubules, and this helps hold everything in place. You can see it here. It's holding that mitochondria in place. It's also little pathways that stuff like roads that certain elements and chemicals are going to pass through as it goes to the cell. It also, these same type of structures are involved in what we call the cilia and the flagella, and those are the things that will move the cell if the cell has these for movement or for sticking or holding on, whatever may be the case. A cell will also have a vacuole. Now, a vacuole is a storage area for water and other substances. Plants usually have a very large one, and we call it the central vacuole. Now, in your cells, you will have some vacuoles that will help store liquid and other things, but the plant has a very large one that you can see. Let's look at a picture of that in just a second. The main job of the animal cell, plant cell, really any eukaryotic cell, you could say the main job is to produce proteins. That's what it does on a regular basis if you had to give it a title. And the proteins that it produces is dependent upon the DNA. So the cell is like a factory. Its product is protein, which go to the body and serve different functions. The DNA has instructions to build the protein. These instructions are sent to the ribosomes. Then the ribosomes build the protein and send it through the endoplasmic reticulum. Finally, it will be packaged in the Golgi Apparatus, where they're completed and tagged to export outside the cell to go to do whatever it does if it's leaving the cell. If it's staying in the cell, we're going to skip a couple steps. We're not going to put it in the endoplasmic reticulum. We're not going to send it through the Golgi Apparatus. We're just going to make it in ribosomes that float around in the cytoplasm. But protein production is key, and that could be everything from an enzyme to a hormone to some kind of signaling molecule somewhere else in the cell to some kind of function like mitosis or meiosis or whatever may be the case. The proteins are the ones that cause these things to happen in the cell. Here we have a plant cell. Now, you can use this comparison back to the animal cell, the previous lecture. You notice that it's got a lot of the similar things that was in the animal cell, except for three important differences. Let's look at those. They have a cell wall. This surrounds the membrane and provides additional support. That cell wall is made up of cellulose. Cellulose is a structure that your digestive tract can't even break down because you don't have the right enzymes to break that cellulose down. But it's a very rigid structure that helps the plant stand up, to have support, to get its leaves out for photosynthesis, things like that. The chloroplasts, which contain the green pigment that functions in photosynthesis, and we'll talk more about this later on. And then, of course, that central vacuole that we talked about, that contains water as well as other things that are contained in there, but primarily water. You can see this once again. Here is that central vacuole, that big open area that's going to store a bunch of water. The cell wall out here on the outside, and then the chloroplast here, and we'll go in more detail with these later. So if we look at a plant cell under a microscope, what you would actually see is because of that cell wall, it has a very specific shape. Most of the time with plants, you're looking at the shape being a rectangle. Okay, it's just a rectangle. And in there, under a light microscope, you'll actually see tons of these chloroplasts. Any one cell could have 30 or 40 chloroplasts in there. They pack chloroplasts inside of these cells like crazy, especially depending on which plant cell you're looking at. But you can see the green dots all floating around in the cell. Those are all chloroplasts ready to make sugar through photosynthesis. There's a couple specific organelles that we'd like to talk about, which are the mitochondria and the chloroplasts. And they're unique in that they have their own DNA. They're about the same size as bacteria, which animal cells are much bigger than bacteria. But we have chloroplasts and mitochondria are about the same size. And they reproduce or divide similar to bacteria as well. When your cell is dividing, the bacteria, or the mitochondria and the chloroplast will literally, for you, mitochondria, not chloroplast, will divide through binary fission and split apart for those cells. That's unique. Other organelles don't do that. This supports what we call the endosymbiosis theory, which states that eukaryotic cells evolved when prokaryotic cells engulfed or absorbed a bacterial cell. So a eukaryotic comes along, engulfs another one, a bacterial cell, and then they start working together. That's symbiosis. They start performing a similar function together. Mitochondria, they're making ATPs. The cell is using those ATPs, and in return it provides protection for that, and it feeds it. So there's a mutualistic relationship that develops. Finally, I want you to look real closely at the cell membrane. We'll go into some more detail with this later. It is selectively permeable. It regulates what comes in and out of the cell and what in the cell and what leaves the cell. It's composed of a double layer of phospholipids with proteins embedded throughout. That's as much detail as we're going to go into now because we're going to spend a whole chapter on the functions of the cell membrane next.